pest and disease attack. They are prone to all sorts of negative things. You just don't choose them, so you are able to cut them off the next generation. Then another advantage is that it reduces the spread of pests and diseases that is associated with the planting material. Now we talk about the disadvantages of selection. First, it is very expensive to practice. Your records has to be there, so it's time consuming. You have to look at it. You have to take your time to look at how they've been performing. Another disadvantage is that it can only be carried out by a specialist before good results can be achieved. A specialist has to be on ground to see, when there is these traits, when you have this, when this is inconsistent, when this is consistent, then this is what it means, this is the interpretation of this that you are seeing, and things like that. So it can only be carried out by a specialist before good results can be achieved. Then the next one is that certain traits may not be able to blend with the soil and climatic condition. Some traits may not blend to the soil and climatic condition, so that's a disadvantage. Then certain desirable traits may be eliminated during selection. You might have even overlooked it. That this particular line is this trait is good, or this trait will have been better, but it might be lost during elimination. You might have eliminated it when you are selecting. Then selection is a physical process, therefore it is not genetic. So it's what you can see, the traits that is bringing out, that's what you see, and that's what makes you to choose whatever you want to choose. So it is not genetic. Then the last one is that it is not suitable for crops which takes many years to grow. Crops that take many years to grow, you can't do selection for them because it is already time consuming, even for crops that don't take long. So the disadvantage is that you can't use selection method for a crop that takes long to reproduce. Now the third crop improvement method, breeding or hybridization. Breeding or hybridization. I've talked about introduction and selection. So breeding is the third one, or we call it hybridization. Breeding or hybridization is the development of plants by transferring heritable quality from one plant to another through the fusion of male and female gametes. When you transfer characters that can be inherited from one plant to another in a male and female gametes, when the male and female gametes fuse together, so that's what is called breeding. You have this character in a male. You are looking for this height, this size, this color. You find it in a male. Then this is the height, size, and color that you have in the female. When you fuse it together, that is what, when the male and female gametes are fused together, that is called breeding or hybridization. It is also a process whereby two plants are crossed to produce a hybrid such that it will have a combination of the parental characteristics. In hybridization or breeding, the resultant of offspring carry the characteristics of both parents. Then the progenitors or the offspring that result from the process of hybridization usually combine many good qualities of the two parents, thus producing a crop that is superior to either of the parents. By the time the offspring is carrying characteristics from both parents, so it is better than either of them. That's just the meaning. The offspring that you now have is better than either of the two of them, even though they were wonderful, they were good. That was why you were crossing them. But the offspring that you now have as a result of their fusion is better than each of the parents, so it's an improvement. Then we have two types of breeding. We have inbreeding and cross-breeding. Two types of breeding. Then in inbreeding is a process by which closely related species of crops are pollinated. They are closely related. That's what makes it inbreeding. They are very, very related. They are closely related. But continuous inbreeding will lead to the development of pure lines or purebred crops. They are closely related, they are almost the same, they are alike. So when you keep crossing them, it will lead to what we call pure lines or purebred crops. The advantages and disadvantages of inbreeding. First, the advantages. Advantages of inbreeding include you have purebred or pure lines being developed. Then another advantage is that the crops that are disease resistant are developed. Then it can also be used to upgrade local cultivars. Those are the three advantages that we are highlighting under the inbreeding method. Then the disadvantages now. Continuous inbreeding can produce weak hybrid. By the time you keep crossing plants that are closely related, you keep doing that down the line, you may discover that you will produce very soon down the generation weak hybrids which will not be able to perform or adapt to the prevailing soil condition and even the environmental conditions. So that's a disadvantage. Then the next one is that since only one trait is emphasized, other traits will be lacking. Then the third one, there is a drop in the yield of the crops in terms of the quality and the quantity. By the time you keep doing inbreeding of closely related crops, then there is a drop in the yield, in the quality, and even in the quantity. Now, the second type of breeding, cross-breeding. Cross-breeding is a process in which unrelated crops are pollinated or fertilized. The crops that are pollinated in cross-breeding are not related, unlike what we have in inbreeding. But the crops must belong to the same species or closely related species. In cross-breeding too, the crop must belong to the same species or closely related species for cross-breeding to happen. Cross-breeding results in the production of an offspring that is of a better or superior quality than either of the parents, and this is called heterosis or hybrid vigor. Cross-breeding called hybrid vigor or heterosis. Then the advantages and disadvantages of cross-breeding. Advantages first. It is used to develop high-quality breeds of crop. So you have a high-quality breed of crop in cross-breeding. Then it can be used to upgrade local varieties of crop. You can use cross-breeding to upgrade local varieties of crop. Then disease and pest resistance crop can also be developed. You can develop disease-resistant crops and pest-resistant crops using cross-breeding. Another advantage is that crops which can withstand adverse climatic and environmental conditions are developed, because you are bringing the best. You are bringing two good parents to cross. So you have a crop or a resultant crop that can withstand adverse climatic and environmental conditions. Then offspring with early maturity periods can also be developed. So you can improve it, developing a offspring that has early maturity period. Then the disadvantage of this crossbreeding is that it cannot be used to improve crops which do not produce flowers, which means it cannot be propagated by seed. It can only be done by flower. So it can be propagated by flower because you are transferring the pollen grain. So you cannot use seeds in this type of breeding. Now, we move on to talk about genetics, because we are talking about breeding, so you have to talk about the genetics. And first, I'll be talking about terms that are used in genetics. First is genes. Genes are hereditary units that are suitable or situated at the loci of chromosomes. They are responsible for transmitting characters from parents to the offspring. So they are hereditary units that is found in the loci of the chromosome. So genes are located in the chromosome, and they are the ones that is responsible for transmitting of characters. So when there is a character that is found in a parent and also in the child or in the offspring of the plant, then it is the gene that is responsible for that transfer. Then the next is the chromosome. Chromosomes are thread-like structures that are found in the nuclei of a cell. You find a gene on a chromosome and a chromosome in a cell. Then the next one is traits or character. Traits or characters are heritable attributes that a farmer may choose. Traits or character things that can be transferred to the offspring. For instance, seed size is a trait or a character, color is a character, the height is a character, disease and pest resistance is also a trait or a character in a crop. Then the next term is zygote. Zygote is a cell that results from the union of the two sex cells, that is the union of the female and the male cells. Dominant genes or character. These are characters that manifest heterozygously. They are the character that is dominating, from the words dominant, that is manifesting, is the one that you can see, is the one that is pronounced. That is what is called dominant genes. Then recessive genes are the ones that is suppressed in the presence of the dominant gene. So dominant and recessive genes are together, and it's like a comparison. If there is no dominant gene, then there is nothing that you can call a recessive gene. So the one that is dominating, that is more pronounced, is called the dominant character, while the one that is being suppressed is called the dominant gene. Then a hybrid is an offspring that is obtained from a cross between parents of different characters. So you bring a parent with this character and another parent with the other character, then the offspring that you obtain from their cross is called a hybrid, because they are not the same. That's why you call the offspring and hybrid. Then genotype. Genotype is a trait that is inherited from both parents. Is usually not physical, is in the gene. Then phenotype is a physical or physiological expressed trait of an individual. The traits that you can see, okay, this is the height of this plant, this is the color, so that is what is called the phenotype. The physical appearance of an individual is called its phenotype. Now we move on to talk about the Mendel's law of inheritance. We are going to be talking majorly about the two laws of Mendel, the first and the second law. Now the first law is the law of segregation of genes. The first law states that genes are responsible for the development of the individual, that they are independently transmitted